let me start on the list of topics that I will discuss here. So, of course, I will start with some basic stuff about microscopy, and then I will switch to quantitative phase imaging and make a comparison with the fluorescent microscopy, because, of course, this is the main part of our job, just to try to avoid and overcome some of the limitation of fluorescent microscopy. And then I will switch to flow cytometry, and I will give some of the, I will tell you about some progresses about the bidimensional cell analysis by QPI, thanks to also to the AI. And then I will switch to tomography by single cell in flow cytometry modality. And I will focus the attention about the needs to develop a specificity for QPI, because, as you know, QPI is not has no specificity like fluorescent microscopy. And then I will show some of the application and tool that we are trying to develop just to manage all this stuff about tomography, the big data, and so on. So let's start with the basic about the microscopy. Let's see something about the history of microscopy and Nobel Prize. If we go in the history, essentially about optical microscopy, more or less, we can mark just 3 Nobel Prize have been awarded about the things. The genius of Fritz der Nike, a very simple idea, just by opening in a filter, he was able just to was an inventor of this phase contrast microscopy that effectively revolutionized the microscopy field, because he made some invisible visible just by increasing the contrast by these holes inside this filter. So in 1971, instead, Dennis Gabor was awarded by holography, by invention of holography, even if he started with, he would like to make a sort of a magnifying lens from electronic microscopy. At the end, holography developed itself along the different routes. And one of the routes, more promising routes is actually the digital holographic microscopy. And then we can mark the third one, that is the 2014, the beautiful development of super resolver fluorescence microscopy. Thanks to this tool, nowadays the humankind can see into biology and to see all the structures inside the cells and around the cells. And the interesting thing is that the Nobel Prize in 2014 was in chemistry and not in physics, even if the microscopy is a branch of physics, traditional. But why it was awarded in chemistry? Because essentially in fluorescent microscopy, as a mediator, a chemical agent. Without a chemical agent, you cannot visualize the things inside the cells. So let me give you some ground basic concept by quantitative phase imaging. Essentially, is a modern branch of the phase contrast microscopy and is closely associated with the digital holographic microscopy. This is a simple, traditional, long time lapse overnight of fibroblasts, and you can see a nice color map in which essentially is it possible to sampling the physical thickness of this object, of the cells. And so in this way, the contrast is highly enhanced because it's possible to calculate the optical path difference. So the optical delay experienced by, for example, by flat wavefront passing through the cell. And so all this delay give you the optical path difference. That is a quantitative measure, physics measurement about the cells. But the problem connected to this kind of things is that, of course, if you make a comparison between fluorescence and QPI, of course, we can say that fluorescent microscopy is, has a high degree of specificity. You can see the color nucleus here, so you can identify without any doubt the presence of the cone. Here, where is the nucleus? Okay, you see some sort of dome and heel here, but you do know exactly where the nucleus is located inside. You cannot measure here because you have overall information that is integrated all the thickness of the cell in this case. So this is the main problem of digital holography and all QPI methods: the lack of specificity. As a difference between fluorescent microscopy. If you go to the basic properties, features of fluorescent microscopy, we can say that is a powerful visualization tool of the cells, offer high sensitivity and high specificity, offer, thanks to the Nobel Prize, amazing super resolution. But the problem is that you have is not free of errors. There are a lot of errors. It's prone to errors also fluorescent microscopy because labeling process can be invasive. Okay, it can destroy the cell, it can cause abnormal cellular behavior. So, for example, if you would like to try to test a drug with a cell, and we are doing this with a fluorescent microscopy, you don't know exactly if the chemistry of the staining is affecting or not the behavior of the cell or the drug instead of the drug. Then also the problem connected intrinsically with the fluorescent phenomena. You can have the laser light, the photons can be toxic for the cells, or even you can have photo bleaching. That means that you can destroy eventually the measurement and so on. And so this contrast agent can disturb normal function of the cell in this case. So just summarizing again, stain-free microscopy is important because you can preserve cell viability, non-invasive, live cell imaging, long-term observation without the risk to disturb the cells, and essentially you can preserve also the native structures and the native morphology, and so you can have also real-time analysis of the process that occurs inside the cell during all the life of the cell. So again, by summarizing the advantages of the single cell for quantitative phase imaging, we can say is a label-free, high sensitivity, quantitative analysis, live cell imaging, and potential application, of course, are very attractive about the cancer diagnosis, infectious disease, drug investigation, and so on. Let me go, okay, if you would like to have more details about the digital log, of course, there are many books, one about this, but let me shift now to another important paradigm about the single cell analysis. This beautiful paper in 2015 about single cell, which is the importance of single cell, because most of the analysis are made on a statistical number or a huge number of the cell, but you risk to lose the information about the single cell analysis. And so if you would like to advance precision medicine or improve disease model and research, it is necessary to shift to the single cell analysis. What can be done at the state of the art currently about the single cell analysis is flow cytometry. Flow cytometry is a tool that is able just to make a probing of a single cell in a different modalities. One modality can be scattering. Is the most famous flow cytometry is a scattering analysis in which you can collect fluorescence or scattering signals from the cell, sometimes side scattering or forward scattering. And the other class, the more advanced, are instead based on imaging flow cytometry. Okay. Of course, if you stay on the left side, you can have high throughput and thousands and thousands of cells per minute. Okay, it is very fast. On the other side, since you have to collect the single image of the cell flowing inside microfluidic channel, of course you can have you should have a much lower high throughput in this case, because you can collect 100 of cells per minute and so on. So typically, in scattering flow cytometry, for example, if you plot the side scattering and forward scattering, this is usually adopted just to classify inside the white blood cell, neutrophils, monocyte, or lymphocyte, because they have different scattering signature depending from the internal structure inside of each one of the cell. In different ways, instead, in imaging flow cytometer, essentially you can staining different things inside the cells. You can stain the nucleus, so you can stain the nucleolus, or you can say that the other sub-compartment inside the cells with different color. Modern systems are able to make simultaneously at maximum, or I think 5 different staining. So you can staining different sub-particle inside, and so then you visualize this part, you can measure this part, you can calculate the density, and so the system can discriminate, and can select the cell and sorting the cells with different things. Together, of course, also with the bright field images. So you have a bright field image, different simultaneous different colors. And this imaging flow cytometry can be applied for immunology, oncology, biochemistry, but also for oceanography, for example, for diatoms, okay, in the ocean, or microbiology, or toxicology, or nanotechnology, interaction, nanoparticle with the cell, and so on. But every time, of course, you need staining. So you need chemical agent or proteins or other things that are attached to the sub-compartment. And so you have to design, you have to make engineering your stain agent, and then you have to start the process for this one. So if you make side by side pros and cons about image flow cytometry, you can have, of course, high specificity, rapid count, cell sorting, subsequent cell analysis, so on. The limitation essentially are based on fluorescent labeling and indirect visualization, because you cannot have access to direct visualization, but you color something and you see the color expressed by this phenomena. So also the process is manually time consuming, is depending from the operator also that make the process. And as a last limitation, all the commercially available imaging flow cytometer are based on 2D analysis. That's it. You can do fluorescence, everything, but just limited to the analysis. This is the way in which, okay, imaging flow cytometry make the scattering plot just to select and sorting different kind of cell depending from the counting of the fluorescent pixel inside the images. So we can say good for specificity, is a very good and powerful tool for single cell, quite good high throughput in consideration of image capture. So you cannot reach, of course, a million cells per minute, okay, for scattering analysis. It requires staining and 2D imaging. Okay. Okay, this is just a repetition of this. So let me see which is the desirable microscope of the future. Probably he should accomplish all this stuff: stain-free, single-cell imaging, 3D, to be quantitative and high throughput. Probably all this stuff, all these features should be inside in the microscope of the future to make all these application in biology, biomedical, point of care, clinical diagnosis, personalized medicine. And of course, to manage all these tools, all these functionalities of this microscope, it is necessary to develop new tools that allow to manage all the data and information to apply this kind of stuff. Now, so we can say also that computational microscopy is an important part of microscopy. Probably 90% now of the new development in biomicroscopy is related, and analysis is related to the computational aspect. And now I can start to show you some of the results about single cell in 2D quantitative analysis by QPI, and also possibility throughput for two specific applications. One about liquid biopsy, and the second one about phenotyping, which is the setup that we use, just a standard, very cheap plastic chip, in this case, in which we can use a microfluidic channel, just the cell with the liquid are injected inside the channel, and we build up a microscope around the microfluidic chip, like this one, okay? And what we see is essentially, this is on the left, you can see the collection of digital hologram that you are able to collect while the cells are flowing inside the microfluidic channel. Maybe you can notice that they rotate during this, while they are traveling across the field of view of the system. And so you can see on the right side the calculated quantitative phase images of quantitative phase maps of this bidimensional quantitative phase map of this of each one of these of these cells. And so we are trying to exploiting the quantitative information inside these 2D images of each one cell, just to make some investigation. The first one, the possibility for a labor-free assessment of drug resistance of, in this case, epithelial ovarian cancer cells, in which we use two different kind of cell. One is chemical is resistant to the chemical agent, okay, and the other one is not is reacting in good way to the drug, and so by standard protocol given by the doctor and so on, we applied this kind of measurement in the microfluidic chip with the 2D phase map, like in this one. So we collected all the images of bidimensional quantitative image you see on the bottom here, and by making an analysis by machine learning of different characteristics of this one, that means size and the thickness and the other stuff correlated to the quantitative maps, we are able to discerning the two different class of cells. This is very important because you can eventually test the drug in this kind of stuff. One more application. Now I have a project financed by Italian Minister for Research. The name is Cyto-Tomography, in which we are trying to develop a sort of personalized radiotherapy for people affected by the cancer. And so we are trying to identify if there are changing in the cell after irradiation by X-ray in different conditions. And so, for example, we estimated the effect again with the same, exactly with the same principle that I showed before for ovarian cancer cells. In this case, we just exposed it by X-ray, and we were able just to sorting, okay, different morphological characteristics that include not only size but also quantitative phase measurement inside the bidimensional phase map, just to discriminate such kind of cells. One more application, phenotyping. Another recent paper in which we tried to phenotyping the different neuroblastoma cell lines. Okay, we said, okay, maybe we can use AI tools with the bidimensional QPI just to try to differentiate and to select a different cell line on neuroblastoma. And so, for example, at the beginning, of course, our aim was to try to start the first step toward the liquid biopsy. That means that you have to search for cells inside the blood samples, and the cells that can be confused with the tumor, with the circulating tumor cell, are monocyte, of course, because they have a similar shape, okay, similar density like cancer cell, or other cells, of course, no red blood cell, okay, that you have to remove from your sample. But in this case, they can be confused. So the first attempt was to try to differentiate from monocyte from neuroblastoma, okay, and then we are with new strategies just to differentiate the different cell line, NB1, NB2, and then Chiak-P212, and then the other exotic neuroblastoma line. And so we were able to demonstrate that by using some strategies by using AI tools, we were able to discriminate just by using it, by processing the dimensional map of the cells in this case of flow cytometry in 2D. Then let me go now instead to switch to 3D, just to show more example for 3D. We started this with the idea to find new strategies for liquid biopsy. Why it's important liquid biopsy? Because sometimes there are some cells that are released by main tumor. They go in the blood, and go in the blood, they can release in different organs and so on. So it's very important to find strategies to detect early this kind of cells, and so for early detection of the cancer, to avoid the spreading of the tumor inside. It seems that this demonstrated that years before the cancer can develop, there are circulating tumor cell going inside the blood system, and so it's important to do this. So the idea was to make blood samples. So you can see cancer cells here. We made some experiment. You can see the cell entering the field of view. They rotate at least one full rotation inside of the field of view. That means that we have information. We have the chance to collect information for different probing direction. Here, the laser the direction is a single direction, okay? While the cell is moving, while it's passing through the field of view. So we have the unique chance just to probing the same cell from different angles. And so essentially, in this way, we can we have the possibility to to make this label-free imaging, flow cytometry, and using deep learning from different stuff. Again, AI, in this case also ovarian cancer cells in presence of unbalanced status and so on. Or one more paper again about classification of ovarian cancer cell by, again, holographic, label-free holographic image flow cytometer, and again with the same strategies in which we try to joining the AI tools with the full the information that we were able to retrieve from the 3D tomograms of the cell. So let me go now to give more details about the switch between 2D and 3D. So we have on the left just an array of pixels. Each pixel has a meaning just to have the integral accumulation of optical delay of the coherent laser beam passing through the cell. On the right, we would like to have a three-dimensional shape of the lens, and we can do this just by means of a tomographic holographic flow cytometer. And the ambition was just to try to find a unique morphological fingerprint in 3D, a unique fingerprint for a single cell. If we succeed in doing that, can open new possibility just to identify all kinds of cells, because we believe that anyway there is this uniqueness of the cell in this case. If such a system would have, of course, many advantages in terms of label-free, quantitative, high throughput, single cell analysis, but the bottleneck is the lack of specificity, because we have a very powerful physical measurement, okay? Because we are using interferometry. We know that interferometry is probably the most sensitive measurement gauge that has been developed by humankind. In fact, it has been used also for gravitational wave, interferometry. And so, in this case, we are using a very powerful measurement tool for measuring the physical properties of the cells. And so the problem is that we have this whole 3D information of the single cell, but we don't know exactly all the voxel inside this 3D cloud of information belongs to the nucleus or mitochondria or whatever. So essentially, this is an example in which, for example, in our experiment, you can see cancer cell from inside. We applied some sort of automatic tracking and identification of ROI, region of interest of the cells. And so this is our procedure to go into this tomography. In this case, as you know, in a standard X-ray tomography, you have the patient at rest inside the X-ray machine, and then you have the X-ray generator and the detector that is rotating around at a fixed angle, okay? And thanks to the angle, correlation between the angle and the absorbance of the X-ray at a fixed position, is possible to retrieve the tomograms, okay, of this object. In our case, the cells are moving, they are rotating themselves, and we are using all the feature of digital holography just to make possible tomography. Why? Because essentially we are using 3D imaging possibility of digital holography, because of course, if you have a cell at a certain position along the optical axis, and then we have another cell at a different position, we can recover the focus. And so we can shift the cell exactly, all the cells, or all the position of the cell exactly at the same position. This is very important. Otherwise, different height of the cell. Thanks to this flexible focus, we can have, of course, we have quantitative phase contrast, and then we developed some rotation angle tracking, precise rotation angle tracking, to make possible the right correlation between the probing direction and the angular position of the cell in a certain instant of time. This is a schematic sketch of the system. In this case, you see red blood cell flowing, is rotating. I have shown before also in the real movie what's happened. Thanks to the different tricks, for example, we are considering, in this case, a red blood cell sort of optical lens. If you consider optical lens, when the red blood cell has a tilting angle, it experiences a sort of a coma. And so if you retrieve the aberration of this lens, you can retrieve exactly which is the angle by the evaluation of the aberration of the cell that is rotating inside the field of view. And this is the typical collection of the phase map. All the phase map now are aligned exactly in the same position, are correlated exactly to the same specific, at a certain specific angle. If you collect all the information, you can get 3D tomograms of this one. This is very important because, of course, you can have a 3D shape, but you can have also quantitative information inside. You have the complete, the full, the whole distribution, for example, hemoglobin inside the red blood cell, thanks to these tomograms, and not an average in standard medical analysis. Of course, you have average on millions of the red blood cells. No, you can have hemoglobin. In this case, you can have a signature at the single cell. You can see here different cuts of that direction and plane for the tomograms in different conditions. But let me go now to the main focus, a challenging focus about the lack of specificity in digital holography and in QPI. Essentially, we can consider three relevant cases in the examples that I'm giving you now. We can have the case A, in which you have cells just adherent to a substrate, okay? And so in this case, you can have quantitative parameters. So you can get some specificity by calculating, for example, dry mass, refractive index of the cells, okay? You can have a second condition. You have a suspended cell, so a cell that is suspended in a liquid buffer, so has a spherical, almost a spherical shape. And this you can collect the bidimensional map of this sphere, and by making the calculation of the quantitative features of this bidimensional phase map, I have shown you that it's possible to get some information, to achieve some information to discriminate the sorting cells, healthy cells from tumor cell, and so on. And the third one case is the possibility to get the tomograms. But when you calculate the tomograms, okay, in typical setup in which you have rotation, you put all the phase map, you correlate with the angle, so you put in a computer, you get this phase map. And this phase map, you cannot recognize nothing. Because, of course, in this drawing, this 3D voxel that you are seeing here, it just was chosen one threshold, just to show one level or two level, just to show some aggregation of the voxel inside. But there is no physical meaning or correlation between the voxel inside this 3D object and nucleus or mitochondria or nucleolus and so on. Okay, and so the problem is, the challenging is to try to extract the right information from this cloud of the three-dimensional voxel of the cell. Let me say, for example, for a cell on a flat substrate, you can collect the shape, okay? You can visualize, or you can also identify also organelle inside the cell because there is a different delay. They are essentially a flat object. This is quite intuitive to extract the information in this case because they are a very thin object, and you can distinguish the information. But if you have a spherical object, it becomes very challenging because you have this sphere that in addition, this behavior like a lens is focusing light also, okay? And so all the information is convoluted inside this spherical object, as it's not easy. So also if you make a thresholding analysis and process of your tomogram in a simple manner, just putting some threshold, you cannot distinguish the organelle inside the cell because the refractive index change is so small from one organelle to another that it is not possible to retrieve such kind of information. But anyway, by bidimensional map, you can get this information by putting some quantitative numbers because of the physics of these numbers, and you can separate. You can select the different cell because you have intrinsic information that you can extract, for example, thanks to machine learning. So in 3D, instead, it's very complicated to extract this cloud of a pixel, which is the physical meaning, which is the physical position of the organelle inside. And so our main job is to try to get from this gray 3D distribution homograms specificity. This is just a cartoon on the right, okay? But we have on the left side the nucleus, the nucleolus, the mitochondria, the membrane, and so on. And we are trying to do this. And for example, if you see many cells, in this case, a neuroblastoma cell, and you set some threshold, you can see nothing, okay? You can have nice information, nice physical, high sensitive information of this cell, but you cannot see anything. So there are two ways until now that has been developed for extracting this specificity for this 3D. One on statistic, that is our work that we developed also from Dimitri here, and another group in Korea that used the AI, okay, to extract information this 3D. This is Professor Park. He started also a company. The name is TomoCube, and so they make tomograms of a single cell, but a static cell, okay, not flowing cell. We are the only one to make a flowing cell tomography. And so he tried to make this deep learning to extracting sub-organelle. And essentially, if you use deep learning, you have to you have to afford infer multiple 3D fluorescent tomograms, refractive index measurement, corresponding subcellular target. You have to training the network. You have to achieve eventually good generalization. Otherwise, every time you have to retraining the network. So it's very complicated because there are many cell lines. They are very different each other and so on. Instead, in our case, we tried to make this statistical analysis in which we were able to demonstrate the possibility to extract the nucleus from this. And so in our case, we achieved the direct extraction of a specific 3D intracellular structures for this flow cytometry mode. We essentially, as a first step, identified the nucleus. The nucleus is very important biomarkers. For example, cytoplasm ratio between cytoplasm and nucleus can be a biomarker for cancer and so on. In our case, this algorithm was not based on AI. We choose this kind of path in bioimaging, labor-free 3D, tomography, flow cytometry, statistical segmentation analysis. This slide will show you which is the basic concept behind this kind of algorithm that was developed thanks to Daniele Pirone, one of the smart researchers in our lab, in which if you start by some hypothesis with this, by considering that the nucleus, for example, is centered inside the cell, and you make this inference analysis statistical, you can create some test in which you are able to identify the number of voxels that belongs to the nucleus and the voxel that does not belong to that don't belong to the nucleus in this case. And so it was tested, of course, on numerical stuff. And then by analyzing the tomograms, the profiler, refractive index, and the histogram, it was possible to build up the nucleus by this statistical analysis to define this. We made some comparison also with the image flow cytometer. And so we had the projection 2D in fluorescence, statistical analysis. It was in agreement more or less with our analysis in 3D. And so we were able, essentially, this is a message, to pass from the line up to the bottom line, in which we were able to now give to a scene some physical and biological meaning to the cloud, because in this case we can represent essentially which is the size and location of the nucleus inside the cells. Then we tried also with the nucleolus, and so we succeed also to do this. We are on the way now to publish a paper also on and to patent again with Dimitri patent a new invention about nucleus, so I cannot give detail now, but essentially, let me go now to the next step. We demonstrated in this Nature Photonics paper, the first single specificity in a cell. So we are trying to make more step forward just to having more organelle, multi-specificity inside. And I will show something. So we choose this budding cytoplasm vacuole yes cells. It's very interesting, very simple cell, in which you can have very easily duplication of the cell. You can have multiple organelles inside the cell. You can have vacuoles. You can have also nucleus, okay? And so you can identify multiple things. You can see here the result of our tomography, in which you can see the nucleus, the vacuole, the blue one, and then the cytoplasm inside. You see the projection in the phase map, the reprojection of the tomograms in the phase map, and you can see how it appear also the fluorescence. It's not exactly the same cell, but you can see in the same stage cell in the same stage of evolution. Okay, and you can see the colors are inverted. So the red one now is the vacuole, and then the nucleus is blue. So, so essentially, this is the cell as it appears in confocal microscope. Of course, by using this budding process, this is a yes cell, we were able to duplicate also two different cells with multiple organelle inside, and essentially we were able to identify here different two nucleus, two vacuoles, two membrane, and doubling the specificity for this one. So at the end, we say, okay, maybe we are trying to develop 3D specificity. That means that we are going in 3D. Probably we cannot use 2D visualization method just to calibrate our system, to understand what's happening, how to assessing the performance, or how to find a new way to assess the performance of our 3D specificity algorithm. And so we decided to go in 3D. I will show here a simple journey. It's just one possibility, one possible journey inside the 3D context, in which an operator can select one of the tomograms here and interrogate the system and visualize with the Google's in 3D environment, just to select in this case the cell, visualize the cell, and also walking through the cell, go inside and to interrogate the system, which is the name of the component, but also to extract the quantitative measurement connected with the specificity of this kind of cell. This is what's for us. We believe that, of course, this can be very useful also, for teaching, okay, for training. But we believe that it can be very useful also for improving such kind of process and algorithm, because in this case you have a 3D tool in which you can judge the progresses in your work in identify subcellular organelle inside a cell. Different stuff for different cells. You can go. In this case, there are some lipid droplets inside. And so you can go, you can evaluate and things, or one more. In this case, we inserted inside the cell, there was an uptaken process of graphene nanoparticle inside the cell, and here are tomograms in which it's possible to visualize the aggregation of tomograms inside. But anyway, as I said, this can be very useful for collaborative research, but also for the education training, but also as intrinsic element for a tool for develop 3D specificity tool. Now, having 3D data, that means that you have a huge of the data, because first one, when we make our experiment, we record 1 minute, 2 minutes, 10 minutes of digital hologram of 1,000 cells passing through the field of view, very huge. Then we selected the field of view, the region of interest, and then we build up the tomograms. And so we have 3D data, so 3D voxel, okay? And it's quite difficult to manage all this stuff. So we say maybe we can start to make some effort to simplify the management of these things, and speeding up, for example, the computational process of 3D refractive index tomograms. This is quite easy. Data compression and management, or classification of cell bases on 3D tomograms, also phenotyping, or making more step forward to liquid biopsy. Let me go speeding up the tomographic reconstruction. For example, of course, we start with digital holography on the left, and we get phase map, bidimensional phase map. Then we put together, we stick all together the information of two-dimensional phase map of a different orientation, and we get 3D tomograms. So we develop a network just to simplify in a quickly the transformation from digital hologram to the bidimensional phase map. One more possibility was the possibility to use some compression strategies for this from this data. But before to give some details, even this work was in cooperation with the Professor Saltis here, I will say why we had the idea to use the Zernike polynomial. Because a few years ago, we started the possibility to use a red blood cell like a sort of a lens. And as you know, lens, in this case, for example, you use a red blood cell just to make imaging, and we gain 20% of a magnification than imaging some scratch on a DVD in this case. And this was because if you look at the discocyte, you can recognize more or less the shape of a negative lens. Okay, if you see a spherocyte, you can get a positive lens. If you have an echinocyte, you have destruction of information because you are scattering different things. Also, the bottom line is taken by high school physics test, okay? And you can see similarity between this kind of lenses and the red blood cells. So this analogy was quite powerful because we made some experiment and calculation, numerical simulation to understand also diffraction pattern from this object by numerical simulation. But we made some experiment by changing the buffer and by transforming a discocyte in a spherocyte. In this case, we were able essentially to get a variation in the focus. For example, you can see that during the experiment of transformation by changing a discocyte in a spherocyte, there is transformation by 2 focal points to 1 focal point. Why 2 focal points? Because the discocyte, if you take the paraxial part of the lens, is actually a negative lens. It is a negative focus, okay, on the left. But if you take the toroidal shape, it has a positive focus. Then, when you destroy this shape by increasing, by changing the shape from a discocyte to a spherocyte, it becomes just a single positive lens. And so you can get just the focus just on one side. And so when you, people familiar with the lens, they know that the people for fabrication of lens, they have to take care of aberration. And how is possible to take care of aberration and to model wavefront aberration of the wavefront at the exit pupil of the lens? Often they are using polynomials to describe the wavefront. One of the most powerful is Zernike polynomial. So we say, okay, maybe since the red blood cell lens, so the cell can be a lens, maybe we can use Zernike polynomial like a model to make a strategy for compression of data. And so we did, in fact. In this case, you can say that by modeling, we discovered that there are also, usually for wavefront aberration, the Zernike polynomial in 2D. But there exists, from mathematical point of view, also polynomial in 3D, of Zernike polynomial. That is very interesting because essentially you can model your lens by Zernike polynomial, and so you can calculate which is the loss of information by sacrificing some resolution information. You can save a lot of space in your computer for data, for space saving up to 95%, but just a negligible information loss less than 1%. And you can see here just the plot to show how, by increasing the number of the order of the polynomial that you consider for modeling your cells, in this case up to the order 25, you can see that you can define the physical characteristics. You can reproduce most of the physical inherent characteristics of the single cell. And this is quite interesting. Now, 2 more examples about intracellular 3D visualization, thanks to 3D tomography. One due to the exogenous nanoparticle, another one to endogenous nanoparticle. Some things that are inside the things. Here, we published this paper on possibility to visualize, I have shown before the graphene here. Here on the right is just a cartoon, just showing the uptake process of a graphene oxide, the graphene nanoparticle uptaken by the cell. And when we look at this imaging of the cell, just the adherent cell or the supercell, it was not clear where are these nanoparticle clusters of graphene? Outside, inside, around the nucleus? It was not clear. And you see, we see this nice decoration, but we were not sure. So we can try. We can try with the tomography to give an answer to the exact location of this one. So by considering different uptake time of this nanographene oxide particle, in this case, you can see small aggregate of the particle exactly located in some portion, so you can have a 3D coordinate of the location of the aggregates of nanoparticle in different conditions. Or if you have huge uptake of nanographene in this case, so you can have also this decoration exactly around the nucleus, along just one diameter, diameter of the sphere. There is this decoration of clusters of the oxide. You can, of course, make some speculation about quantification, distribution, this nanoparticle inside the cell. 3D visualization instead of endogenous intracellular particles, lipid droplets. You know that lipid droplets are very important about cellular mechanism, about their dynamic role, and they provide defense in infection and other things, also in cancer and different things. And usually, TEM imaging or fluorescence microscopy are the elective tools to visualize such kind of the cell. By means of tomography, it's possible to get information about 3D distribution and to have a nice indication of presence of this lipid droplet and the location. Of course, we are limited by resolution of the optical microscope, of course, numerical aperture and so on. But you can make a nice evaluation of this liquid droplet in 3D in label-free mode. I will jump this because I have a few slides at the end. Liquid biopsy by 3D flow tomography, again, the same scheme that I shown before. And again, we made some, we continued some investigation about 3D in this case. I have shown before neuroblastoma in 2D case. Now all the analysis and process has been made in 3D. And again, I will skip all the details about the strategies and the tools and details about but essentially we were able, thanks to the neural network, to separate monocyte from the tumor cell and then classify different tumor cell line in the same sample that flow inside the things. Phenotyping, of course, again by 3D. Instead to make 2D analysis, we switch to 3D and we improved a little bit the analysis of this resistant cell in different conditions. Here, summarize more or less. Now, I think we can apply tomography also to diatoms, okay? And we foresee exploitation of many possibilities of application for liquid biopsy, personalized medicine, drug testing, and so on. I think we have more than 10,000 atlas of the cell, different cell line, different things. We are continuing to accumulate information on this one, and probably we can produce an atlas for virtual reality also to show and also eventually to sell like Netflix on demand for the people that would like to do. The last shot that I would like to say, we are working thanks to a project funded by Regione Campania, the region where Naples is located, is the possibility to segment lysosome for rare disease. Lysosome has a problem for rare disease analysis. You can get this kind of information, and you can see in wild type and in genetically modified mouse, you can have accumulation. That is meaning that there is a disease in this case, accumulation just in one hemisphere of the things. And now we are working. It is interesting to show you side by side comparison between confocal, that is claimed to be a 3D, and flow cytometry holographic tomography. And you can see the difference. Of course, so you can get information by confocal in 3D, but it's not exactly the same way, because essentially what do we claim here? I will show here. That we were able to identify some biomarkers, original new biomarkers, just by calculate the solid angle, okay, portion occupied by the lysosome in this case. And this is completely new in respect to what can be done by confocal analysis, in which you can have also the distribution of lysosome for adherent cells is perturbed by the physical mechanical adhesion of the cell there. So you cannot be sure exactly that the accumulation, which is the percentage of accumulation and so on. And here you can see that this is was nice. We are submitting a paper about this in these days, in which you can see that we applied a sort of a cycle drug. So wild type, knockout, then we use a drug just to recover the disease, and we observed the shift of the lysosome from hemispherical distribution to the general distribution with different drugs. In this case, NPC1 for knockout and other things. We were able to demonstrate this kind of accumulation and evaluate also the degree of this accumulation, just extracting some things. So some acknowledgement about the project and collaboration, of course, and here the project, the current project that we are working in Italy. This is my group, there's Pasquale Memmolo, Lisa Miccio, Vittorio Bianco, Simonetta Grilli, Volodymyr Tashenko. We have a collaboration about microfluidic with Unina, Zewang, Conne De Natale, Daniele Pirone. Now he's a researcher. He was announced in his career from postdoc to researcher, and then Marica Valentino and Giussi Giuliano. And we are located in Pozzuoli, 10 kilometers north of Naples, there. And I will show something that maybe can be of interest, just that is related in some way to holography of fringes, interferometry. We are located here. You see Pozzuoli there, and you see that a logo of a mountain there. It is a volcano. The volcano is Solfatara. And we are in Naples. And essentially, I live, okay, so I live here, and this is my lab here. Okay, and if you see, you can see from Google Map several craters here. So that suggests that in the past, very ancient time, there were many volcanoes here. And in fact, that was the case. And here again, you can see that essentially the institute is here. Here, Isasi, Cenel. And here you can see Solfatara. This is the volcano here. Okay, and it's interesting because you see this is beautiful map. From one side it's beautiful, from the other side is horrific, because that means that we are in a very dangerous piece of land. And I will show why. This is a synthetic aperture rather, and that are wrapped phase. You cannot wrap like in our hologram, in our interferogram. It's really nice, and that means that it's horrific because there are several cycles, so in millennia, okay? And now the last cycle started in 2005. And you see that the uplift from 2005 to now is like a child growing, okay? A teenager from 0 to 138 centimeter, and of which 20 centimeter just from January of last year, 20 centimeters. So all the soil is going up. And this is technically it is named Mar. Mar means is a sort of volcano, very ancient former volcano that was created by lava with cold water. Okay, and they have this typical very teeny and narrow stick here, and so all the experts they don't know what's happening there. There are a lot of sensors, a lot of investigation study, but still there is no information. But of course, you can see smoke, you can see the smell of sulfuric acid. Also for the building having canteen on the underground for CO2 accumulation. There is also this possibility. Of course, it can be dangerous. And so essentially, this is the evolution in millennia. So the caldera, the big one, you saw there. And so there was Santa Maria delle Grazie 4,500 years ago. And then Caldera di Agnano. And then now here, this dome here, just on the top here now, there is the Academy of Air Force of Italy. Exactly there. It's a beautiful place because just you can see, look, all the Gulf and Pozzuoli, Ischia, Capi and something. And now the most recent in Solfatara is there. That is exactly this. We are here. It is interesting that this is the curve, the experimental curve of the uplift during the years, and you can see also the variation in height of the water. And so, if you start, because it's interesting, because there is a temple just in front of the airport, and you can see sign of the water along the millennia because of corruption action of the animals or in the water or the water. And you can see that this excursion was really true along the millennia. And so this map, what is this? It's just an anomaly in the total deformation. That means that recently in a paper, they demonstrated that essentially this part is under-deformed. It was not expected. And so we had the last earthquake was just 16th of February, and the magnitude of Richter scale 3.8, and we have about 1 centimeter uplift more. Thank you very much.